Good day to everyone on the line, and welcome to JDAC's Building a Smarter Lab, How RFID and Machine Vision Improve In Vitro Diagnostic Testing webinar. My name is Justina Valencia, and I'm part of the marketing group at JDAC North America. On the line, we also have our co-hosts, Kelly Holt and Annika Matas. Today's agenda will consist of a quick intro to JDAC, an overview of IVD testing, current challenges, and applications and benefits for both RFID and machine vision and in vitro diagnostic testing. We'll conclude the webinar with a Q&A session at the end of the presentation, so if you have any questions during the webinar, please feel free to type in your questions into the Q&A window to your right so I can read your questions to Kelly and Annika at the end of their presentation. Please be sure you're writing your questions to me, the host, Justina Valencia, to ensure your questions are read. So a little about JDAC and why we are an expert on this topic. JDAC, which is a business unit of Novanta, specializes in machine vision, RFID, barcode, printing, and computing products, providing both off-the-shelf and optimized product solutions for medical device manufacturers. Our expertise is in healthcare, and in fact, about 90% of our business is with medical device manufacturers. As for our presenters, Kelly Holt is our Machine Vision Product Manager. She brings over 10 years of experience in medical device product management and is certified in machine vision. She also received her MBA from the University of Florida. As for Annika, she brings over 10 years of experience in the data collection industry, which includes barcode printing, personal identification, and RFID. She received her master's in systems engineering and MBA from Loyola Marymount University. And now I'd like to hand the mic off to Kelly Holt, who will take you into a brief overview about IVD testing. Kelly, take it away.
Thank you, Justina.
Yep.
Good morning, everyone. The FDA has defined in vitro diagnostics, or IVD, as tests done on samples taken from the human body, such as blood or tissue. These tests detect diseases and other conditions and are used to monitor a person's overall health to help cure, treat, or prevent diseases. There are three phases in the IVD testing process. The first phase is the pre-analytical phase. The second phase is the analytical phase. And yes, you guessed it, the third phase is the post-analytical phase. Let's take a look at some of the steps included in each phase. The pre-analytical phase covers all the steps from the patient assessment until the actual test is conducted. This includes selecting the proper test, preparing the patient, collecting and transporting the sample, as well as all of the steps necessary to prepare the specimen for testing. During the analytical phase, the actual test is conducted, and the results are interpreted and reported during the post-analytical phase. And as we all know, additional tests may be ordered based on the results. Therefore, this process can become a continuous loop. Oftentimes, clinicians and laboratory personnel rely on clinical analyzers to complete much of this process. These analyzers are able to perform many tests with minimal human involvement, thereby allowing laboratories to process many more samples without comparable increases in staff. Now let's take a look at some of the current challenges with IVD testing. The vast majority of errors in the in vitro diagnostic process happen in the pre-analytical phase. The most commonly reported pre-analytical errors include the missing sample or test request, wrong or missing identification, sample contamination, or inappropriate containers. Unfortunately, these errors have an impact on the patient. Misdiagnosis, delayed treatment, redraw a sample, and lengthening of hospital stay can all result from these pre-analytical errors. And according to one study, these errors are also costing the hospital to the tune of $1.2 million annually based on a 650-bed hospital. In addition to errors in the pre-analytical phase, there are issues inherent in the IVD process, including transportation and storage delays and mistakes. Mechanical issues can cause the wrong test to be performed on a sample. And finally, variation in human analysis is a common IVD pain point. To take you through some of the applications and benefits of using RFID in IVD testing, I'll hand you over to Annika.
Thank you, Kelly. Okay, so to get started, let's look at how RFID can help to solve some of these pain points. RFID is not a new technology. It's been around for many years. And if you're familiar with the Gartner hype cycle of innovation, it has actually finally passed the phase of disillusionment and is now reaching that wonderful place that's called the plateau of productivity, with increased adoption in many industries. Retail and manufacturing are leading the way. While adoption in healthcare is still lower in comparison, we do find RFID gaining traction in several application areas, such as product identification and tracking, asset management, as well as access control. Let's look at those areas more closely. The main areas where RFID can improve IVD testing include secure access and staff identification. Most hospital staff members already carry an RFID badge to gain access to different rooms in the hospital. That same badge can be used to access equipment or perform specific tests. And the benefit of using RFID is a higher level of security and therefore improved traceability. The second application area is regular equipment maintenance. RFID can be used to monitor equipment usage, and that coupled with data about NTDF with regards to that specific analyzer can be used to generate predictive maintenance alerts, as well as track when maintenance events have actually taken place. This will mean that equipment and instruments are performing at their optimal level and ensure accurate test results. The next application area is specimen tracking. As Kelly mentioned earlier, a significant amount of waste occurs due to mismanaged samples and tests. RFID enables the tracking of specimen samples in real time or near real time from the point of collection to when the specimens and samples reach their destination in the lab. This helps to reduce errors and thereby costs, ensures accuracy in the test data, and provides us that critical traceability for billing and record keeping. RFID can also be combined with temperature sensors and other sensor technology to record that samples have been kept refrigerated, as an example. The next key application area is identification, authentication, and tracking of consumables that are used in the IVD analyzers. IVD equipment and consumables are sold to healthcare providers, and we always trust that they have the right intentions. And the same is true in the pharmaceutical industry. However, the FDA estimates that 80% of the active and bulk chemical ingredients in U.S. drugs originate overseas. And this increasing global production and supply chain creates vulnerabilities. Without sufficient oversight by the industry and regulators, it can put patients' lives at risk, and it can put test results at risk. As a result, we're facing some startling statistics. According to the World Health Organization, more than 700,000 deaths worldwide are caused by the sale of counterfeit medicines. This is triggering a heightened sense of urgency to track authenticity of pharmaceuticals and laboratory supplies. Many IVD tests are using highly proprietary cartridges and reagents, so the risk in the lab may be lower than the general pharmaceutical space. But it is an area where RFID can help to provide additional protection and information. In addition to ensuring authenticity, RFID can help to monitor the usage and simplify the user experience for using these complex analyzers. Related to my last point about simplified user experience, since RFID can automatically detect what is installed in the analyzer, that information can be used to automate settings such as pressure, flow rate, as well as operating time for a specific test. This, of course, reduces the risk of human error and improves our outcomes. While RFID has a lot of benefits and potential application areas in IVD testing, I do want to state that Barcode is still a very important, viable, and a low-cost option for automation in the lab. In most cases, an either-or choice, I would say, is the wrong way to look at the problem. Both technologies have their individual benefits, and they can complement each other. Here are some of the key situations where RFID is an ideal complement, or in some cases, a substitute for RFID. Firstly, when it's not possible to guarantee line of sight between the ID of the item and the scanner. That's when RFID is really strong and very useful. Second, when we need to read many items quickly or simultaneously. Here, again, RFID brings a lot of value. When we need to update the information on the item itself to note things like chain of custody, monitor and record the temperature or usage of the item, RFID brings the ability not just to read the item information, but also store information back onto the RFID tag. RFID can provide a higher level of security to authenticate either the person holding the credential or the authenticity of the item itself, going back to the point of helping us combat counterfeits. And it can help to make decisions when we don't have connection to a central database. So it gives us a more autonomous solution. Once we have identified pain points that RFID can solve in the IVD lab, we then need to choose the right RFID technology for the specific application. RFID is a broad term, and there are different technologies that fall under that umbrella. Today, we're only talking about passive RFID, which does not require battery-operated tags to identify the location of items. The fact that these are passive RFID technologies means that all these tags derive their energy needed to operate from the antenna's RF signal. In the passive RFID space, there are three broad technologies to choose from: ultra-high frequency, UHF, high frequency, which includes NFC in that space, and low frequency. UHF offers the longest range, but it has low level of individual item discrimination and has poor performance around liquids and metals. There are new tags entering the market that allow UHF to be placed on metal items as well as near liquids to compensate for this. Both HF and LF have a much shorter read range, from contact to a few inches. Low frequency, or LF, in healthcare is primarily used in access control applications. It has a large install base of building door access readers, especially in the US. So therefore, low frequency employee badges are very common. HF is ideal for identifying individual items at a short range, and it offers a series of other features in the tags, such as counters to allow us to monitor usage of an item, as well as encryption to make sure that we have genuine and authentic items and genuine and authentic credentials. So as you can see, selecting the right RFID technology is a deeply intertwined process. And it's one of the key components of that selection is selecting the right tag. As I speak to customers in the field, this is one of the key areas where people are looking for more information as they're trying to choose, especially between UHF and HF in the IVD space. Making the decision requires a thorough comparison of the pros and cons of the two different technologies, understanding the needs of your specific process, such as read range, and the constraints of the environment, either physical space or materials located around the item that you're trying to read. This table here is intended to be directional and to give you an overview of different variants and options. For HF, I have chosen NXP chips to illustrate cost ranges and capabilities, but there are other options available from other chip vendors, so this is by no means an exclusive or exhaustive list. To get us started on defining that range, small memory tags in the HF space that are ideal for item identification can start at about 20 cents. This is by no means a super high volume quote. That's something that you can easily find when you shop around. And high memory tags that can store a lot of usage data and parameter information can store as much 8K of memory can cost significantly more. We're talking about 40 to 50 cents even. The iCode tag is one that I've seen commonly used for consumable identification, as it operates based on the ISO 15693 specification, and so therefore it has a slightly longer reading range than the other tags that I've listed here, which operate in the ISO 14443 specification range. For UHF, general purpose tags start as low as 10 cents, and these are typically used for specimen tracking. Most UHF tags have a specific memory bank for EPC codes. EPC stands for Electronic Product Code, and it's typically 96 bits of writable memory. If more memory than the EPC section offers is needed, certain ICs do have extended user memory, which is typically about 512 bits or 64 bytes. But there are also some high memory tags in the UHF space with either 4K or 8K of memory. Advanced read range tags can cost 20 to 40 cents, and specialty tags that have designs uniquely meeting the needs of placing the RFID tag either on metal or on liquid items can be between 50 cents up to $1. And of course, the more unique your needs, the more varied the cost ranges become. So with that, I hope that you get a good overview of how RFID can be used in the IVD test space and how it can meet different challenges that the labs are facing. Next, Kelly is going to talk to you about machine vision in the same way and how that can be used to help solve the pain points of IVD labs.
Thanks, Annika. There are a variety of ways machine vision can be used to improve IVD testing. By applying software algorithms to a captured image, machine vision can read barcodes, detect vials or caps, as well as determine the liquid level of a sample. Let's take a few minutes to examine some of the machine vision applications for IVD testing. Machine vision can be used for barcode analysis. In the top example, machine vision is used to read 5 barcodes at once. To save time, we programmed the algorithm to look in specific areas of the image for the barcodes. In the middle example, a barcode is wrapped around a vial. By spinning the tube while capturing multiple images, then stitching the images together, machine vision is able to read the barcode, calculate the liquid level, or find a break in the label for sample analysis. And finally, the bottom image showcases a poor quality barcode that was printed directly on a customer's product. A basic barcode decoder could not decipher the barcode. However, by applying a custom machine vision decoder, the barcode could be read and the customer did not have to dispose of the product with the poor quality barcodes. Another example of using machine vision to improve the IVD process is vessel and cap detection. In the first example, multiple machine vision tasks are completed with one acquired image. The barcode on the rack identifies which rack is being placed into the analyzer, and the barcodes on each individual vial are also being read. Each cap and tube dimension, shape, and color can be identified, and machine vision can also recognize if the tube or cap is tilted or even if the cap is missing. The image on the top right shows a variety of caps used by a particular laboratory. By using machine vision, this laboratory was able to reduce the error of color identification and increase throughput by relying on a proven machine vision algorithm to identify the caps instead of the human eye. The bottom image demonstrates 5 vials that are all used in the same clinical analyzer. Machine vision is able to identify which vial is in position, allowing the robotic arm to navigate without damaging the vials. Machine vision is also able to count objects, which are referred to as blobs. As shown in these images, blobs can be pills, microorganisms, or cells. Machine vision can be trained to count almost anything, including the absence of items. Another example of machine vision being used in IVD is processing microtiter plates. A microtiter plate, as seen in this image, has 96 wells. Machine vision is able to identify a microtiter plate, verify its orientation or alignment, and even confirm presence or absence of fluid in each individual well. This slide demonstrates the presence-absence functionality of machine vision. The image on the right is a rack containing vials, but the rack is not full. Without machine vision, a robot would have to check each position within a rack for a vial. With machine vision, the absence-presence of each position can be communicated to the robotic arm, allowing the robot to go directly to positions within the rack that contain vials. The image on the left highlights the empty positions within the rack with a red circle. Liquid level measurement is becoming more and more important in the IVD space. Machine vision is able to calculate how much fluid is within a vial. This is critical for ensuring enough sample is available to complete the requested test. In the picture of the vial with the orange cap, the machine vision software knows to ignore the yellow rack and only focus on the vial. Additionally, algorithms have been applied to the liquid level measurement to calculate for the meniscus. The fluid level is also important to ensure the pipette has drawn enough fluid. This is especially important when the blood components have been separated, which we will see on the next slide. Using color measurement, machine vision is able to verify a sample has been properly centrifuged. When combined with liquid level detection, Machine vision can direct the pipette to the level needed to extract the correct portion of a sample. Additionally, machine vision can detect blood serum quality or index and scan for blood clots. This concludes the presentation portion of our webinar, so I'll hand the mic back to Justina.
Thank you, Kelly. For anybody that has questions regarding RFID or machine vision, please feel free to type in those questions so Annika and Kelly can talk to you guys. So with that, we did receive a few questions, so I'm just going to go ahead and read these aloud. Kelly, I believe this first question is for you, and the question is, can the same camera that reads a barcode be used for machine vision?
Thanks, Justina. And yes, the same camera can be used to read a barcode or use machine vision. The camera actually is just acquiring the image, and it's the software that determines whether a barcode is deciphered or other machine vision applications are executed. JADAC's machine vision software includes a barcode decoding application.
Awesome. Okay, I believe the next question is for Annika, and that is, can multiple RFID technologies be used in one application, HF and UHF together or LF and HF together?
Okay, thank you. Actually, what I might do to clarify that point is go back to a slide. So let me take, or if you give me back control of the presentation.
Yep. 1 second. Let me get that over to you. Okay.
Thank you. So you will help to look at this as I'm answering this question. The answer is yes, you can have multiple technologies used in a single application. There are both tags as well as encoders available that can cover more than one of these technology spaces. Most often, I would say, we see ultra-high frequency and high frequency used together when we're trying to provide staff members access to a specific room and identify them for access to equipment, but then also, give them the ability to sense their presence in larger areas. So specific access to an item and more location awareness applications, so UHF and HF combined. The similar way is where low frequency and high frequency are used together. I mentioned that low frequency is very common in hospitals to give employees access to different rooms and doors. So a lot of the door readers that we see in hospital buildings operate in the low frequency range. But high frequency is a newer technology, and it's great for use for secure access to specific devices or cabinets. So a lot of credentials have a combination of low frequency and high frequency. So two different chips and antennas inside the same ID badge. And that are typically the combinations that exist, LF and HF together or HF and UHF. But it really depends on the needs of the specific application.
Thanks, Annika. Okay, we have another question for you, Annika. It says, does the Novanta software combine the temperature history from data loggers with the RFID tracking, or is that handled separately? For example, are RFID tags automatically associated with a temperature based on their location?
What I'm going to do is actually take that question and review that with our engineering team to have a more precise answer about, from a software perspective, what the techniques are to combine information from data loggers with the RFID tags. So I will owe a specific answer. I want to make sure that we give the right guidance on this question.
Oh, okay, no problem. I will make sure I connect you with the person that asked this questions offline post-webinar.
Thank you.
Yep. Another question for you, Annika, is what are the regulatory or certification implications of adding RFID to an IVD analyzer?
RFID is a radio technology, so IVD equipment that includes an RFID reader is considered an emitting device, and therefore there are some certification impacts of including RFID in the device. However, as we're trying to make that choice, some things are put in place to make it simpler for IVD device manufacturers. Encoder modules can come with modular certifications, which means that the reader that you embed in your device already has a self-contained certification that makes the IVD analyzer certification simpler. You don't really have to deal with a radio certification component. But if you choose a module that doesn't have a modular certification, maybe has been pre-scanned, so it's known to meet the levels of FCC and other regulatory agencies, you will still need to go through those certification processes and make sure that in your development timeline you account for that. It depends on the country. It can take from somewhere to 4 to 6 weeks for FCC in the United States, but in a different region like Brazil or China, the time frames can be 8 to 12 weeks, and the costs vary by country as well. FCC again can be around $10,000, but different countries can have different cost implications for radio certifications. Thanks, Annika. Just want to remind to all our participants to make sure you're typing your questions to me, Justina Valencia, the host, so I see them and so I can read them aloud. Kelly, I believe this next one is for you, and that is how many machine vision applications can be completed on an image?
That's a great question. And pretty much the sky is the limit. Once an image is captured, machine vision can quickly analyze multiple features within that image. Let me see if I can find an image here real quick. So look at this image on the bottom right of the screen. In this particular image, you can
Kelly, I think, sorry to interrupt, I think Annika has to, yep. I believe, yep, there you go.
Can you see it now?
Yes.
Perfect. So on this image on the bottom right, you can, machine vision can obviously identify the vial as well as its orientation. And you would be able to identify the cap. In this case, it's the purple cap, or you could even identify absence, presence of a cap. You would be able to identify the plasma, which is the top level, the kind of yellow color, and conduct a liquid level measurement or a color analysis of that plasma. And you could also identify the red blood cell portion, which is the bottom red in the vial. And again, you could do a liquid level measurement or even color analysis. And even in this one image, you could also do an analysis for clots. It would be able to look at the entire image. So really, any discernible feature within the image, you can use machine vision to look at it all at one time.
Awesome. Thank you, Kelly. I believe this next one is for Annika. When barcode and RFID is used, does barcode and EPC memory ID typically match? What is the most common barcode standard used in IVD? There is a big push to commonize the use of EPC standards or GS1 standards in the healthcare space as well. But it really varies. There is no single standard at this point yet. But I would say that from the market, there's a big push to being able to apply a single and unique identifier to every single item.
in the supply chain, both from a barcode as well as from an RFID perspective. But what you can do is ensure in the process of encoding that the EPC encoded in the RFID tag matches the barcode that's printed on the top, because of the flexibility of the RFID tags and the ability to read and write data into the EPC memory bank.
Okay. If there are any other questions, now's the time. I'm just going to give people a minute to type in any questions you may have. Other than that, we'll conclude the webinar. So I just want to give everyone the opportunity to ask any more questions. Okay, I'm just going to go there. So that basically concludes our webinar. At this time, I'd like to thank everyone for attending our webinar, and a special thanks to our presenters, Annika and Kelly, for their presentation. A recording of this session will be automatically emailed to those who registered for the event within the next week. I just want to add to that in alignment with GDPR, we are unable to send a recording of this event to our European registrants. So if you are in Europe and you would like to receive this recording, please email us at optin@jdactech.com indicating we have your permission to send you marketing information. Or you can visit our website, jdactech.com. Within the next week, the webinar will be posted on there. If any of our attendees have any further questions on machine vision or RFID, we invite you to visit our website, jdactech.com, or schedule a tech day with us by emailing us at techday@jdactech.com for a free technology consultation. Thank you everyone again for attending today, and have a wonderful day.